Welcome back, friends. In this video tutorial we are going to talk about biofertilizers. Okay, so let me write. We are going to talk about, yes, biofertilizers. Okay, now what are biofertilizers? As the term suggests, you can see bio means fertilizers made up with biological organisms. Simply, fertilizers made up with biological organisms. Now, what do we mean by biological organisms? Usually, biological organisms means here in this case we means for bacteria or other agents. Now, we usually stick with bacteria in this case. Now, the type of bacteria that we can use for biofertilizers will depend upon what we need to achieve using biofertilizers. Now, as the term suggests, fertilizers it is definitely having the uh, importance of fertilizers, right? So what are the importance of fertilizers? Now, important features about the fertilizers are, first of all, the most important feature is that they must increase the nitrogen content, right? Nitrogen content of a soil. So that is what we actually want to achieve. We need to increase the nitrogen content of the soil. And for the increase of the nitrogen content of the soil, we need to add different fertilizers, which can be of chemical and all the other types. But nowadays, we are focusing on making the fertilizers with bacterial cells. Now, how could a bacteria act like a fertilizing agent? That's the biggest question of all, right? We need to answer this. Now, for understanding this concept, we need to learn a basic concept of biology that is called symbiosis, right? So, let us talk about symbiosis. Okay. Now, what is symbiosis? Symbiosis is simply the relationship of two different organisms in ecology or in ecosystem. Relationship of two different organisms in which case both of the organisms are benefited, right? For example, say a plant is there. So, let's say a plant is there, and obviously a bacteria resides in the root region of the plant, in the rhizosphere region of the plant. So a bacteria lives there. Okay, now the plant supplies, now in this case both of them will be benefited, right? So here in this case plant supplies all the nutrients to the bacteria, some major nutrients to the bacteria. On the other hand, bacteria need to supply something to the plant, right? Bacteria are very tiny things, how it can supply something. Now in this case bacteria plays some important role of fixing nitrogen from air into soil. Now, as this bacteria fix nitrogen into soil, now the soil becomes nitrogen rich, right? So that's what actual goal of ours, nitrogen enriched soil. Now, in this case, soil become enriched with nitrogen content. Now, plant require nitrogen for their growth. Remember, for the plant growth. So, for a plant to grow, so a lot of writing now. From a plant to grow, what we require, major three different things: nitrogen, phosphorus, and potassium. These three important things are most required things. Now in this case, nitrogen is the most important one. And in this case, we can see some bacteria can fix nitrogen into the soil. Now the plant can be benefited by taking this nitrogen from the soil, right? So that's why it is called the symbiotic relationship. Now in this case, we can use this type of bacteria to fix more and more nitrogen in the soil, right? Now what we actually do using fertilizers, we use chemical fertilizers. So all the chemical fertilizers that we use in those cases to increase this nitrogen content, that's our actual goal. But now in this case, what we can do, we can increase this nitrogen content using some biological agent like bacteria. So that's the actual goal of biofertilizers. Okay, so what kind of bacteria we can choose for making these biofertilizers? Definitely we need to choose for those bacteria. So we need to choose for those bacteria. Let me write it here. So here, okay, so bacteria that are used usually those one which can fix nitrogen, right? So not all bacteria can fix nitrogen. We all know that, because atmospheric nitrogen or N2 is a very tough thing. We cannot fix this directly. None of our higher eukaryotic organism can fix this nitrogen. Only few bacteria and few protozoa and a few type of organisms can fix them. And among them, some bacteria like cyanobacteria. So this is our first goal about the cyanobacteria. right? So let's write cyanobacteria is a type of bacteria which can fix nitrogen, and obviously there are bacteria like endophytic bacteria. So let me write endophytes. Okay, endophytes are also examples. So what are endophytes? I'm going to tell you later. Now these are the major two types of bacteria that we can use. Now remember, I've told you before that there are some bacteria present in the root of some plants. They can fix nitrogen. Now usually this type of plants having this type of nitrogen fixing bacteria in their root or rhizosphere region are called as leguminous plants. So they are called as leguminous plants, right? Now there are other plants which cannot fix, which does not carry bacteria which can fix nitrogen in the soil. There, this type of plants are also there, and this type of plants are present in higher percentage. Actually, they are called non-leguminous. They are called non-leguminous type of plants. Now remember, there are some bacteria present in leguminous plant that can easily fix nitrogen. Now in those case, those bacteria, the example of this kind of bacteria is obviously cyanobacteria. Some amount of them. Obviously, there are some not cyanobacteria. There are some other bacteria of example of that are present in leguminous plants. Example is rhizo. So the example is the bacteria is called, okay, rhizobacter. Okay, because they are present in rhizosphere, that's why they are called as rhizobacters. Okay, and obviously. They can fix this nitrogen for this leguminous plants, but there are also other bacteria present which are usually present in non-leguminous plants, which does not fix nitrogen in that amount, which are called endophytic bacteria, right? Previously it was thought that only leguminous plant or the bacteria present in leguminous plant can fix nitrogen, but nowadays the concept is changed because they have also found some bacteria present in non-leguminous plant which can also fix nitrogen. They are called endophytes or endophytic bacteria. Oh, okay. And the third kind of bacteria that we are going to talk about is cyanobacteria, which are not a symbiotic bacteria because they are free living type of bacteria. So this third kind is different. They are kind of free living bacteria. So they are kind of free living, sorry, free living type of bacteria in this case. Okay. And rest of them are symbiotic. So both of them are here in this case symbiotic type of bacteria. Okay. So we are going to see how we can use this type of bacteria and to produce this biofertilizer. So let's move on. First, for example, the cyanobacteria, and mainly actually whenever we try to produce biofertilizers with all these bacteria, they become really easy because the actual process of producing biofertilizers is almost same for all this type of bacteria. But if we try to make biofertilizers with only cyanobacteria, there are slightly difference from each other, but more or less major pathways of production is similar. So let us look at the production of biofertilizers. Look for production of biofertilizer, or how we can produce the biofertilizers. Okay, so let's look for this. So for this first step of this production of biofertilizers, we must produce our source, right? So source in this case, in this case is obviously uh, what we know as the culture or starter culture, right? Which can be made up with, say for example, we need to produce this fertilizer with the cyanobacteria, for example. So we need to have a starter culture of cyanobacteria. So in a test tube we will be having the starter culture of cyanobacteria. So here are having the cyanobacteria culture. Okay, so now what we can first do is that we can transfer this culture into what is called into a broth. So here it says a broth is there. Now this is a liquid broth. In this liquid broth we are transferring all the culture materials here. So we'll be transferring this cyanobacteria to this broth, right? So now it is becoming liquid. Now after this transfer, we need to take this bacteria out of this broth, and we need to culture it and place it into giant chamber, which is also called the fertilizers, right? So the giant chamber is there. Inside the giant chamber we'll be putting our uh, culture obviously our inoculum, which is here cyanobacterial inoculum, and there are also medium. Now the medium that we obviously provide in this case, most of the time they are made up with soils and sometimes some other things, but usually they are made up with soils and water, obviously. And we put our bacteria. So let's say this green red thing is our cyanobacterial mat. So we'll provide. After some time, it will develop into this huge and large cyanobacterial mass here. So now what we can take this mat out of it. So let's do that. So we take this mat out of it. So let's say this is the cyanobacterial mat. We produced it, we isolate it from the fermenter, and we take this mat, we clean it. So the important step of cleaning, and clean this mat, and then we sterile this mat so that other contamination cannot be present there. Then we take this mat, we degrade it, we break this down and we dry it. So this is about the processing. Drying of this mat must be done. After the drying, we serve this mat as a powder, right? As a powder in this case. So that's how we can produce this kind of biofertilizers. So first we need to make the starter culture, which is made up with the type of bacteria with which we want to produce this fertilizer. Then we transfer them into the broth, then finally transferred. So this is the second step. Then we finally transfer into the giant for the fertilization. So we must write it here. This is where the fertilization is going on. Fertilization means simply the development. So let me write it as a development. Development of all this bacteria is actually carried out in this chamber. Then when the mat is developed, we take the mat, we clean the mat, sterilization purpose, then we dry the mat and we make a powder, and we'll serve this powder to mankind. So that's how we can produce the biofertilizer. Now if we are going to talk about biofertilizers in a specific type, like this cyanobacterial biofertilizer. So after producing this mat, remember, when the mat is going to be produced, we can do two important thing with that, right? So what we get from this mat, we get a lot of cyanobacterial culture. So all of this process are involving the actual goal of is that to produce lot huge amount of something. So in this cyanobacterial starter culture we are having, so let me write, we are having cyanobacteria in less amount. But now what we do, using this process we are increasing the percentage or concentration of our cyanobacteria, and the concentration that we get after this process is as a shape of mat, right? Because there are a lot of filamentous structure are there between cyanobacteria, so we get a meshy structure. Okay, now we can use this kind of mat into the field in two different ways. For example, now what we produce is simply the culture. Now in this powder we will be having simply cyanobacterial powder, right? So we can simply apply it into the soil, but it is difficult because if this is a powder, the application will lead to the loss of many cyanobacteria, because if we just spread it into the soil, it will not work. So what will happen actually, if this is a soil for example, and if we provide this powder onto the soil, so it will be just placed onto the upper surface, but that will not solve our problem. We need to inject, or need to add this powder inside, right? We need to add this powder inside this soil, right? So how can we do this? How can we achieve this process? For this reason we can follow two different important steps. And what are the two steps I'm going to talk about? Okay, so let's say here we are having our uh, say cyanobacteria culture. So yes, say here is cyanobacterial culture. Okay, now we can do two different things. Once we produce the cyanobacterial culture in mass amount, we can code seed with it. So the first strategy that we can go for, it's called the seed pelleting strategy. So let me change the color. Yeah, it is called seed pelleting strategy. Now in this strategy what we can do, we can cover a seed using this cyanobacterial mass, right? For example, let's say here it is a seed, so let me take a green color for denoting the seed. So if this is a seed reproduced, and obviously say the cyanobacterial culture here it is in red, so we simply coat this seed with our cyanobacterial culture, and then this is called the seed pelleting, and we can serve this a lot of this kind of pelleted seed, and the seed will be implanted into the soil. When the seed is implanted into the soil, what we get, so if suppose again, if this is the soil, seed is implanted into the soil, and then the tree will come out and it will be heavy, right? And as this seed is coated with cyanobacteria, it will fix enormous amount of nitrogen there, so soil will be enriched with nitrogen and our goal of biofertilizer will be achieved. Similarly, there is a second technique, and this technique is called carrier mediated inoculum or inoculum carrier technique. So let me write it here. It is called inoculum carrier, right? Now, what do we mean by this inoculum carrier technique? In this case, what we produce this cyanobacterial culture, right? So again we start with this process. So there is a lot of culture. So lot of culture that is just generated by cyanobacteria. Now, what we do now, we take this culture and we attach this culture with some kind of carrier. So here it comes the attachment of this culture with some type of carrier. After attachment into some type of carrier, what we produce? Carrier culture complex, right? So a carrier culture intermediate. Now, using this carrier culture complex, we can apply this into the field directly. Now, what kind of carrier we must choose in this case? Remember, we need to apply this culture into the soil. So soil can be a type of carrier. If we want to use, we can use soil as a carrier. But remember again, what kind of carrier we use, that must be sterile, right? So if you use soil, it must be sterile soil. So usually the carrier we are going to use in this case, let me tell you here. Usually it is used as peat. Peat is used as a carrier. Peat, remember, is a type of coal. It is not found in India, but in not found in India, actually not in use in India, but in other countries peat is used. But now in our country like India and Sri Lanka, Bangladesh, in this kind of countries, if we want to produce biofertilizers, we can use sterile soil, right? Sterile soil, we can use it. Now, what is the role of this carrier? Remember this culture, when we mix this with peat, so it will form what is called a strong like. So peat is taken out, and say peat is very rough thing like that. We degrade this into smaller fragments like that, and then we add culture. So let's say here the culture is in red. So we add culture. So culture is coating all the peat structure, so it will mix with the peat. So this will allow us to handle our culture pretty easily. So that's the basic goal of using it, because this will allow us to handle, so it will help us to handle the culture very well. Otherwise, we are dealing with the biological culture, it will be difficult for us to handle it as it is. So we need to handle it better way. So that's why we're using this kind of carriers. So these are the two ways after the production of it. So remember, whatever process we have seen and discussed is simply due to and this can be carried out. So if this all this process that I have discussed can be carried out only after the development of a typical inoculum, right? So we must produce this inoculum first, then we can use seed pelleting technique or carrier inoculum carrier technique to use it, right? Now usually, now let's talk a little bit about the bacteria that we use in this case. Usually the bacteria that are used, so bacteria in use. So let's talk about that. Now the bacteria we use for this kind of production, usually if we use cyanobacteria, so if we use cyanobacteria, we can go for say Anabina, Baena. So this is Anabina, mind the spelling, and obviously Nostoc, and this type of cyanobacteria can be used for the production of these biofertilizers. And obviously if we use some endophytes, right? If we are going to use some endophytes, we are going to use uh, acetobacter, and obviously azospirillum are important choice. So let me write it here: acetobacter and obviously azospirillum, right? So this is a type of bacterium that we can use as endophytes. Now obviously if you want to use other type of symbiotic relationship, we can use azola, which is also a symbiotic relationship between Anabina, right? So if we use azola, we must use a combination of Anabina and azola, because they are having in symbiotic relationship. both these cases, right? So in future video we'll be talking about how to produce a biofertilizer using only cyanobacterial material, and we are going to see the detailed procedures on how to produce a peat and how to culture them and all these stuffs. But that's it, and I hope that's helpful. Thank you.